Right, so we're going to demonstrate how to look at the rate of reaction of enzymes. And we're going to use catalase, which is produced by yeast. As I said, you can get it produced by liver. Great little experiment you can do. If you cook liver and then drop some hydrogen peroxide on it, nothing happens. But if you put hydrogen peroxide on raw liver, it bubbles away. And that's because the raw liver has, and the enzyme's denatured by cooking it. Okay? So that's a really simple practical, but we're going to actually measure the amount of gas produced. We're using hydrogen peroxide, which in here is breaking down into two OH dot radicals, and then those radicals are then broken down or basically got rid of by turning it into water and creating hydrogen gas. It's really important it does that because that will mean that the yeast can get rid of free radicals. Free radicals will attack proteins, DNA, and so on. So we've measured out exactly 4 grams of yeast. Exactly 100 millilitres of water. I'm going to add that. Again, it's not going to dissolve because it's a cell, it's just going to be a suspension. So when I'm mixing it, I just want to create a homogeneous solution. Ideally, I'm going to use a smaller beaker than this, but I haven't got a smaller beaker. But we don't really want it to go up the sides. So I'll try and get it off the sides there. But the idea is you want it equally distributed. That's what homogeneous means. It means equally distributed throughout the whole mixture. And each time you use this, mix it, because each time you use it, okay, it might just settle to the bottom. So make sure you're mixing it each time you use it. Now to help, because otherwise we'll need more pairs of hands than the octopus has, we're going to use some clamp stands to make sure everything's nice and stable. So we're going to clamp our conical flask, 150 milliliter conical flask. I'm just going to clamp it in. Be careful when you're clamping it not to clamp it too tightly, otherwise it will break. Lower it quite low, to about here, just giving yourself an extra pair of hands. If you don't clamp it, it could tip over because it's not that stable. Okay. We're then going to take our measuring cylinder.
Onion.
The water that I've used here is not, this doesn't need to be distilled water. The water in here is distilled. This water can just be tap water. And we're using the gas displacement method. Keep your books well away from this because they'll get lots of water around, but just put your hand over it, turn it over, put it into the water, let it go. And if by magic the water stays there, and then the water will be pushed out by the gas as it's produced. Take your clamp stand, and again, just to give yourself an extra pair of hands, clamp it in, but make sure the scale is pointing so it can be seen. So if you do the scale pointing between the gap of the things, you can still see it. And you want to just lift it above so it's just under the surface. You've got a delivery tube. Now this isn't necessarily the best way to do this. Okay, there is a better way of doing this. So what will happen is the reaction will go on in here, and this will go on to there. But can anyone think of the better way of doing this? Think of a problem. So let me just set it up. So we're going to take...
You've got to put that thing on that really quickly.
Yes, you've got to put the lid on very quickly because gas will start to escape. So another better way to do this is to use a sealed apparatus with a syringe where you can put the syringe in and you can inject and it stays sealed. So that would be an improvement for this by having what we call a side arm flask, which basically is a flask with an arm coming out the side where you can attach to the hose, and then a syringe that can go into the top where it's all sealed and you can just push it in and it will stay all sealed so you won't lose any of the gas. We're using syringes for ease rather than accuracy because they're not accurate, okay? But today we're just using it for a bit of ease. They go up to 20 mils. So we're going to do the first one. You should always do the purest one first. We're doing the most concentration of enzymes because we've got the most concentration of yeast. So I'm give it a stir and make sure it's stirred up, and then take 20 mils here. Now, the first experiment is going to be 20 mils of yeast. The second one, you're going to take 15 mils of yeast and then take up 5 mils of distilled water, then 10 and 10, and then 5 and 15. And then finally, you're going to do a control with 0 amount of yeast. So no yeast at all, just water. You can tell me the purpose of that. What's the purpose of doing a control? Yeah?
So you can compare it.
So you can compare, so you've got a point of comparison to make sure the hydrogen peroxide isn't breaking down its own. It does break down its own a little bit, that's why you can see it bubbling, but not very fast. So I'm going to put the yeast in here. Again, this isn't the most precise way of doing this, okay, but it's just easy. Yeah, the yeast is alive. It can survive being dehydrated. Now, the amount of hydrogen peroxide you use, you can test, because you don't want to use so much hydrogen peroxide that you get too much oxygen produced. So I'm going to suggest you start with 4 millilitres of hydrogen peroxide. And we're going to measure the amount of gas produced every 10 seconds, because our idea is to create a rate. And not just any rate, we are going to create a rate that is the initial rate of reaction. That is the rate of reaction when it's at its fastest. Do you remember what the graph looks like? When we did this in the previous lesson, it goes up in a kind of vertical linear way, and then it tails off. Why does it tail off? Until it plateaus. Why does it do that?
Because the enzymes denature.
Not the enzyme denature. Nothing's going wrong with the enzymes here. Okay, something to do with the hydrogen peroxide. Yep. Yeah, so it begins to get used up, and so we call it substrate limiting, because not all of the active sites are being used, and eventually the substrate runs out. But we only want the rate of reaction for the first part, so we have to plot a graph of the whole experiment over 120 seconds. You're going to do this over 120 seconds, every 10 seconds for 120 seconds, and then you're going to do it at the other concentrations. We are going to pull the data as a class, like we did before, so we're able to do that. And we'll do that next lesson. So I'm going to take up 4 millilitres. Again, this is not the best way of doing it, but it is the most efficient way of doing it, because it allows me to inject this quickly into here. Now this is where you need to work as a team. Now, I'm not going to do everything here because I haven't got enough hands to do it. But what you need to do is you need to make sure you've got one person who's got the stopwatch and is looking at the... so one person who's got the stopwatch, one person who's looking at this. The person with the stopwatch needs to call out every 10 seconds, say now. The person at eye level, to avoid parallax errors, needs to look at the measuring cylinder, and it will go quite fast. So he needs to write it down. And that, of course, is a point of what sort of error would that be if you're not able to... Yeah, human or what's random error. So check your length is long enough, okay, to make sure it's close enough that you can actually put it in. Have it all set up ready. The person who's doing the measuring is probably also the person who should inject it, because they can inject it and then get down to eye level. The other person could press the stopwatch. But again, to make it fair, what do we need to do? We need to make sure we put we start the stopwatch at the same point. So we're going to start the stopwatch as we put in the bung. Okay. So I'm going to inject it, but in the bung as quickly as possible. As soon as the bung's in, I say start. Okay. So I'll go 3, 2, 1, go. Put the bung in, start, and it's popped out. Okay, this one's a bit short. Okay, but it will continue to produce the gas, and then every 10 seconds, okay, you would measure it. In actual fact, I could lower that down. I would have to discard this one because this one didn't work. Okay, but you can just lower that down to give yourself a bit more space. Okay. And then make sure that's tucked up in there like that. But again, this one is too short. I think we'll get another longer one. Okay, but it will produce the gas. Okay, and the gas will be collected in there. And then every 10 seconds, you'll measure the amount of gas produced, the volume of gas produced. Then you're going to plot that in a graph for each one at the end of doing the practicals. You've got 5 experiments to do. Okay, you're working in pairs. Risk assessment, the hydrogen peroxide. What's the risk?
Don't get it in your eyes.
Yeah, it's dangerous to eyes. It's corrosive to eyes. It's corrosive to skin. And the avoidance is goggles. Okay, and wash it off of your hands if you're getting it on your hands. Okay, in stronger solutions you'd wear gloves, but it's not too bad. And then the emergency, okay, is to inform the teacher, and then we'll go take you to the eye wash, and then you'd go to A&E. So don't get it in your eyes. Make sure you're wearing goggles at all times. Make sure you're standing up, and make sure you keep all of your, apart from a piece of paper you're taking results in, make sure you keep all of your stuff away from the water, because you will get it wet. Okay, great. Let's get started.